In this explainer, we're going to break down a recent discovery published in the journal Nature Aging. It details a molecule that appears to form a crucial link between the metabolic state of an individual cell and the overall health of the entire organism. What this research suggests is a previously unknown signaling system, one that works across different tissues to help regulate healthy aging.
This really is the central question that stems from this new line of research. The study we're about to explore presents some compelling evidence that such a molecule might actually exist, and that it could function not only as a biomarker of health, but also as a potential agent of longevity itself.
So, to really understand this discovery, we'll walk through it in 5 parts. We'll begin with the concept of healthy aging, then introduce the messenger molecule at the core of the study. From there, we'll examine the key lifespan experiments done in mice, connect those findings to human health, and finally, map out the molecular pathway that seems to make it all work.
Let's start with a bit of scientific context. You see, in aging research, there's been a major shift in focus. It's not just about extending lifespan, that is, the total number of years a person lives. The goal now is more about improving what we call health span, the period of life spent in good health without the chronic diseases and disabilities we often associate with aging.
Now, the very foundation of our health is communication between our cells. This constant coordination is what maintains metabolic balance across the whole body. But as we age, this communication can start to break down. We've known for a while about sensors like AMPK that monitor energy status inside a cell, but a much bigger question has lingered. How do different cells and tissues all over the body coordinate their metabolic states with each other? This study proposes a novel answer.
That brings us to our second section, where we meet the molecule at the center of this story. The study focuses on a protein called CtBP2. It's a well-known metabolic sensor, but as it turns out, it seems to have a function that was previously and completely unexpected.
So what is CtBP2? Well, its established role is inside the cell's nucleus. In there, it acts as a sensor for the cell's energy state by reading the balance between 2 molecules, NADH and NAD+. A high NADH to NAD+ ratio signals a high-energy metabolic state. When CtBP2 senses this, it changes its shape and helps regulate which genes get turned on or off. The key point here is that, until now, its function was thought to be entirely confined within that single cell. It was viewed as an internal manager, not an external messenger.
To understand CtBP2's newly discovered role, we first have to talk about exosomes. Think of them as tiny biological parcels that cells release to communicate with one another. They're like an intercellular mail system. These little packages travel through the bloodstream and other fluids, carrying a cargo of proteins and other molecules to distant cells, effectively delivering messages and materials across the body.
This quote from the paper really captures the core finding. The researchers discovered that CtBP2, this protein they thought only worked inside the nucleus, was being actively packaged into these exosomes and sent out of the cell. And this wasn't random. It happened specifically when the cell was in a state of high metabolic activity, the very condition CtBP2 is designed to sense. This was a paradigm shift. CtBP2 wasn't just an internal sensor, it was an external signal, broadcasting a cell's healthy metabolic status to the rest of the body.
The study then went on to detail the actual mechanism for this secretion. It happens in 4 main steps. First, that high-energy state inside the cell activates CtBP2. Second, a specific type of enzyme tags CtBP2 with a small protein marker. Third, this tag acts like a shipping label, directing the CtBP2 to be loaded into newly forming exosomes. And finally, the cell releases these exosomes out into circulation. What's also important is that the researchers found this whole process is hindered by oxidative stress, a known driver of aging, which suppresses the enzymes responsible for tagging CtBP2.
So we have this interesting mechanism at the cellular level, but the really important question is, does this secreted CtBP2 have any meaningful effect in a living organism? That's what our third section is all about, taking these observations from the cell to a living animal.
The experimental design here was quite direct. Researchers took aged mice, 2 years old, which is quite old for a mouse. They then gave them one of 2 types of exosomes. The main group received what they called C+ exosomes, which were loaded with CtBP2. The control group received C- exosomes, which were identical except they lacked CtBP2. This setup ensured that the only significant difference between the 2 groups was the presence of that one protein. The results of this experiment were quite notable. The aged mice that received the CtBP2 containing exosomes showed significant improvements in key markers of health span. As you can see from the data, their grip strength was higher and their endurance on a treadmill was greater compared to the mice in the control group. These are standard measures of physical function in aging mice, and they point to a direct link between getting exosomal CtBP2 and an improvement in health.
The impact wasn't just on health span, though. The study also tracked survival. It found that the administration of CtBP2 exosomes was associated with a 60% reduction in the risk of mortality for these aged mice. This indicates a statistically potent effect on longevity.
Of course, these findings in mice immediately bring up the next logical question. Is there any evidence that CtBP2 plays a similar role in humans? Our fourth section explores the data that connects circulating CtBP2 levels to human health in the aging process.
To look into this, the research team developed a method to measure CtBP2 levels in human blood samples. They analyzed serum from two distinct groups: people from families with a known history of longevity and people from families with more typical lifespans. The result was a clear correlation. Individuals from the long-lived families had significantly higher levels of circulating CtBP2.
Further analysis revealed a consistent pattern. First, in the general population, serum CtBP2 levels were found to go down as age went up. Second, lower levels of CtBP2 were associated with a higher rate of age-related cardiovascular diseases. Third, in people with diabetes, lower CtBP2 correlated with more severe complications. And finally, smokers, who are exposed to high levels of oxidative stress, had significantly lower CtBP2 levels than non-smokers, which provides a strong link back to the cellular mechanism we talked about earlier.
This brings us to the final piece of the puzzle, the how. Now that we've seen secreted CtBP2 can extend health span in mice and is correlated with human longevity, the researchers wanted to understand exactly how it produces these beneficial effects in the cells that receive it.
The evidence suggests that when an exosome delivers its CtBP2 cargo to a recipient cell, that protein isn't just a passive passenger. Instead, it seems to act as a catalyst. It actively kicks off a specific chain of molecular events, a signaling cascade, that ultimately changes the cell's metabolism for the better.
So let's walk through this proposed signaling pathway. It begins when the delivered CtBP2 activates an enzyme called CYB5R3. This, in turn, shifts the cell's energy balance, increasing its NADH2-NAD+ ratio. This high-energy state then activates the recipient cell's own internal CtBP2, which then partners with a protein called FOXM1. This pair then increases the expression of a gene called PAICS. And finally, the protein made by that gene produces a molecule that is a known direct activator of AMPK, a master regulator of metabolism and a well-established target in longevity research.
And as this final quote from the paper highlights, the pieces of this pathway are already well known in the field of aging. Independent studies have already shown that activating CYB5R3, FOXM1, and especially AMPK can extend both lifespan and health span in model organisms. The real novelty here is the discovery that exosomal CtBP2 appears to be the upstream trigger that unites all of these protective proteins into a single coordinated body-wide signaling network.
So, what this research provides is a comprehensive model stretching all the way from a molecular mechanism to organism-wide effects and finally to human correlation. It positions CtBP2 as a circulating signal of a healthy metabolic state, one that in turn promotes longevity pathways in the tissues that receive it. This leaves us with a forward-looking question. If lower levels of this molecule are indeed a biomarker for age-related decline, then perhaps measuring it could one day be used to assess biological age. And looking even further ahead, it raises the possibility of developing therapies aimed at boosting this natural protective pathway to improve human health span.